day to day, it's always problem solving. You do that individually, but more importantly, you do that as a team. And that's fun because it gets my gears going. When we work every day, it's smaller problems that we're trying to chip away at, but then they're very big questions that we also answer. My work focuses on breast cancer, and that affects about 50% of the population. As a woman, we know folks that are affected by it in one way or another. So I wanted, even if it's a tiny bit, make a little bit of a difference in understanding the disease.
I have spent my whole career trying to understand the process of metastasis, which is the ability of cancer cells that spread from the initial cancer that appears in a patient. Most patients do not die from the initial cancer. Sometimes 20 years later, they develop the secondary tumor. Then it's a problem because they start disrupting the normal function of those organs, which are vital. Our goal is, why do we wait until that manifests? Our goal is, let's catch it earlier. We try to understand during the evolution of cancer, what is the source of these cells that behave in this dormant phenotype across the body. And we have developed the models and molecular readouts to pinpoint these dormant cells, to actually intervene and try to attack these cells and eliminate them, which are commonly lethal.
We are very interested in how cancer develops and progresses, not from a genetic standpoint, but actually from an epigenetic standpoint. And that means the changes in how our genome is interpreted and how gene expression changes are controlled in cancer progression. We've been interested in some very challenging problems, one of which is a problem called drug resistance. In cancer, there may be, for example, genetic mutation that drives the tumor growth. And there are therapies that will target that specific mutation and target that signaling pathway to shut it down. The tumors are very smart and they find a way around the drug and they find alternative pathways to keep growing. We took another approach to understand if there were some epigenetic changes that were actually allowing the cells to work their way around the drug. We found that there are indeed epigenetic layers involved in creating this drug resistance in cancer cells. And so we could think about ways to target the epigenome to prevent this drug resistance from happening.
My lab at Mount Sinai is interested in identifying new targets in oncology. We study a family of methyltransferases, which are enzymes. So enzymes are proteins that catalyze different functions in the cells, and we are trying to identify those that are not necessary for normal cells, but they're very important for cancer cells. And so the idea is that we can selectively target and kill cancer cells by targeting these enzymes.
So the focus of my laboratory is to study liver cancer biology. So liver cancer is one of the most frequent tumors throughout the world, and also is one of the most lethal. We focus mainly on understanding different therapies that are being used in patients and how those therapies act, and also how we can choose those therapies and give it to the right patients. We found that there was a gene that basically was preventing mutation to respond to immunotherapy. So instead of giving them immunotherapy, probably we should give them something else, or maybe a combination with immunotherapy so that they can become responsive.
I'm not the same person that I came in 5.5 years ago. I'm a better scientist. I think I'm a better communicator. I'm a better team player. But also, I am an example as a woman of color. Not a lot of people get to be in a place where I am. So it's almost like an obligation for me to come and be visible, because I want other people to say, hey, I can do that too.